Hello, welcome to another tutorial on data-driven biology. In today's session, we will explore the basics of differential gene expression analysis and introduce a powerful and user-friendly Python package called DEGA. Let's begin with the fundamental question: what is gene expression? Gene expression is the biological process through which the information encoded in a gene is converted into a functional product, either an RNA molecule or a protein. This process is central to the central dogma of molecular biology, where information flows from DNA to RNA to protein. In practical terms, the abundance of mRNA in a cell reflects the activity of the cell or the specific function or activity of genes. So, what do we mean by differential gene expression analysis, or DEGA? It is the process of comparing gene expression levels between two or more biological conditions, such as treated versus untreated, healthy versus diseased, or wild type versus mutant. By analyzing expression patterns, we identify genes that are significantly upregulated or downregulated, helping us to understand what type of changes occur at molecular level between two conditions. DEGA is a powerful tool in modern biology. It allows us to understand the disease mechanisms, identify potential biomarkers, and study cellular responses to treatments and discover novel therapeutic targets. To interpret DEGA results efficiently, there are several key concepts we need to understand. First one is expression counts, whether it is a row counts or normalized counts. Row counts refers to a number of sequencing reads mapped to each gene. While they provide a direct measure of RNA abundance, they are influenced by factors like sequencing depth and gene length. To correct for these biases, we use normalized counts. Common normalization methods include the CPM, that is counts per million, TPM, transcripts per million, RPKM, that is read per kilobase million, or DSeq2's size factor normalization approaches. The goal of normalization is to enable a fair comparison across different samples or conditions. The second important thing is the fold change, or log2 FC. Fold change represents the ratio of gene expression between two conditions. We use log2 transformation to make the data more interpretable. For example, log2 fold change means there is a twofold increase in the expression. If the log2 fold change is equal to -1, means it is a twofold decrease in the expression. Using log2 scale helps maintain a symmetry around 0 and simplifies the interpretation of up and down regulated genes. Third one is the p-value, or adjusted p-value. P-value indicates whether the observed expression difference is statistically significant. However, with thousands of genes tested, multiple comparisons can lead to false positives. This is where the adjusted p-value or false discovery rate comes in. To control for false discoveries, methods like Benjamin-Hochberg for FDR and the Benferroni corrections for family-wise error rate are commonly used. The main aim is to reduce the false positive rates while identifying truly significant genes. The fourth important thing is the data visualization. Given the vast number of genes analyzed, visualization are essential. Two commonly used plots are volcano plots, which highlights both the significance and magnitude of expression changes. Then heat maps, which reveals expression patterns across samples. These visualization tools help to interpret complex data or results at a glance. A typical DEGA workflow consists of experimental design, data acquisition, pre-processing and quality control, normalization, statistical testing, interpretation and visualization. Here are some widely used tools for performing DEGA. R packages, there are DC2, HR or Lemur. Then there are web-based applications like Galaxy, IDEP. Then Python-based tools like DEGA, which we will explore in this tutorial. In the rest of this tutorial, we'll walk through how to use DEGA, a Python-based package tailored for differential gene expression analysis. Now let's start our tutorial on DEGA Python package in Google Colab. As we know, the Colab provides an excellent environment for package handling without needing for local installation. So all the steps and the instructions will display on the screen. Just follow from top to the end. Step 1. Set up your environment. First, open a new Colab notebook. We'll begin by installing the DEGA package. It is important to ensure compatibility with NumPy. So we'll specify an older version. In this case, NumPy 1.24.4, which is known to work well with DEGA. Step 2. Import required libraries. Once installed, let's import the necessary packages. Step 3. Load sample data. For demonstration purposes, we'll use the sample dataset provided in DEGA's example notebook. In real cases, you would use your gene count metrics and phenotype metadata file. Step 4. Explore the data. Let's take a quick look at both datasets. It is important to note that the columns of gene counts must match the row indices phenotype data exactly. Phenotype data has a shape of 21 samples and 10 attributes. Step 5. Create DEGA dataset. We will now create a DEGA dataset using the strain column as our variable of interest. This tells us we have 28,907 non-zero read counts across 21 samples. Step 6. Run the analysis. Start the analysis using the following command. This might take a few minutes depending on the data size. Step 7. View summary results. Once the analysis is complete, let's generate a summary of the results. This will provide the number of upregulated and downregulated genes. You can extract and save these areas for downstream analysis. Step 8. Visualization. MA plots. MA plot. We can highlight genes of interest by specifying them directly in the MA plot. This allows for easy identification of specific differentially expressed genes within the overall expression landscape. Volcano plots. Sample correlation plot. The sample correlation plot generates a sample-to-sample correlation heat map, enabling us to visually assess similarities and differences between samples based on the expression of significantly differentially expressed genes. This is particularly useful for detecting outliers or verify sample clustering. Gene correlation plot. Similarly, a gene correlation plot can be generated to explore how different genes co-vary across samples, helping to identify potentially co-regulated genes or gene modules. PCA explained variants. Using the PCA explained variants plot, we can evaluate the cumulative variance explained by each principal component. This helps determine the number of components that best capture the underlying structure of the data. Principal component contribution. We can also visualize the contribution of each principal component to the explained variants individually, helping us to understand which components are most informative. Principal component visualization. Principal components can be plotted to reveal sample clustering and separation, often reflecting experimental conditions or biological variants. Co-expression network. With the co-expression network plot, we can visually evaluate the co-expressed genes based on correlation patterns. This is useful for identifying gene modules that may be functionally related or co-regulated. Dispersion plot. The dispersion plot illustrates the relationship between mean expression and dispersion for each gene. This diagnostic plot is key to evaluating the accuracy of dispersion estimation in the model. It is a critical step in RNA sequence analysis. A sparsity plot. A sparsity plot provides insight into the distribution of expression values by plotting the maximum to sum ratio versus total counts for each gene. It helps identify genes that are sparsely expressed or highly sample specific. This concludes our tutorial. Thank you for watching and goodbye until we meet again with another meaningful and insightful lesson.